Factor VIII is the rarest factor in blood. There's the smallest amount of it in total protein and as numbers of molecules. It took a long time to purify because you have to take a lot of blood to start with. We ended up using batches of plasma that were from around 1,000 donations of blood from individuals that were pooled, and we processed through a series of steps that isolated the factor VIII away from every other element in blood. The key to this was the advent of monoclonal antibodies, which just became available as a tool around the time I started on a major effort to get factor VIII completely pure. The reason to want it completely pure was so we could carry out the biochemical analysis of a protein, which involves breaking it down to establish the sequence of amino acids, starting at the N-terminus and working on through. It wasn't possible to sequence right through factor VIII. It turned out to have 2,332 amino acids in a single chain in the mature form, albeit with a 19 amino acid leader sequence. The complete fitting of that together took a major effort in sequencing fragments, and we did complete this, but we confirmed it from the cDNA, that's the RNA sequence, and from the gene sequences. Now, the total effort to do this took my team at the Royal Free Hospital from 1978 to 1982. And during that time, we worked our way through about 10,000 litres of blood. We were able to use two monoclonal antibodies. One was to the carrier molecule for factor VIII, which circulates in the blood fairly tightly bound to the von Willebrand factor molecule. Sorting out the difference between the deficiency of von Willebrand factor, which gives rise to von Willebrand disease, and the deficiency of factor VIII, which gives rise to haemophilia A, was one of the outcomes of this work. But by using the carrier molecule as a target to pick up the complex, we could then get a purification that took us about a quarter of the way to pure factor VIII. And then subsequently, when we'd got rid of the von Willebrand factor, we could grab hold of factor VIII using a monoclonal to factor VIII itself that we'd been able to raise using the partially purified factor VIII. So this was a stepwise bootstrapping exercise. And when we had a column of antibody to factor VIII, we could elute from it entirely purified factor VIII. It was as pure as we could detect. There was no other contaminant visible on a very sensitive silver staining gel. And over the course of 24 hours, we could go from a kilogram of crude cryoprecipitate, coming from about 1,000 donors, to a single sample of 1 milliliter, a teaspoonful, of eluting buffer, which contained 1 milligram of factor VIII with about 4,000 units of activity. That was the starting point for the major exercise in sequencing, which led on to the cloning and ultimately the synthesis of factor VIII. My team at the Royal Free was myself and 4 other scientists, Francis Rotblat, Donna O'Brien and his brother, and Alison Goodall was the maestro who made the monoclonal antibodies. Over at the team at Genentech in California, which was one of the first of the new biotech industry companies, there were about 40 people working on the project, amongst whom I must mention Dick Lawn, who was the maestro who did the libraries from which the clones were made, Gordon Vihar, who's the protein biochemist who took our pure protein and broke it down and then established the short sequences of amino acids in peptides, and Jane Gitcher, who worked out the sequence of the gene and how that was divided up into its exons and introns. Also, Bill Wood was working on isolating the messenger RNA in a copy form and finally establishing that that could be used to synthesize active factor VIII, the first synthetic active factor VIII. And that was finally achieved in about 18 months from start in 82, when we gave them the pure protein batches, through to 84. And when that was announced, we had reached the goal that had been a target for at least 50 years. So you can imagine we were fairly elated. It led on over the next 6 years to the production of the recombinant protein that could be used in patients. And the clinical trials started in 1990 and were highly successful using the synthetic product, which took over eventually as the major treatment, although blood-derived factor VIII is still used for some purposes. For example, it's useful for eliminating antibodies if those develop. But the breakthrough point was really the day when a cell line was producing synthetic factor VIII for the first time in early April 1984.